So in the interest of time, we need to move on to the next speaker, and it's my privilege to introduce Dr. Richard Scheines from Carnegie Mellon, who will speak to us on computational causal discovery. Thank you.
Well, thank you for having me here. It's my training is in philosophy and computer science, and it's incredibly interesting and helpful to watch people confront real cases and have to make real decisions. So while we're waiting for the slides to come up, I can just give you a very, very quick perspective from which I'll try to come. Instead of looking at any particular case studies and any particular methods for individual cases, I'm going to try to look at, in the most general sense, a set of methods for identifying plausible candidates for causal relationships, which then have to be assessed using all the tools you've heard about today in other contexts. So I show unabashedly a picture of Pittsburgh here because I think causality is worked on all over the world, but in computational causal discovery, Pittsburgh is becoming the center of the universe. So the first thing I want to say is to understand what I'm trying to get at here, I want to contrast what we're doing with a different sort of activity I refer to as estimation. So let me give you an example of each, and so that'll help you, I think, understand the difference. So here's a question that came up and was answered, or at least attempted to be answered by an article by Miguel Hernan, a colleague of Eric's in Harvard in the year 2000 with several other people. And it was, what's the effect of this drug, zidovudine, on survival among HIV-positive men? And there were all the problems I think you have seen today. There was confounders, lymphocyte counts, and not only were they confounders, they vary over time, and they were endogenous because they were going to be actually affected by the treatment originally, and what the treatment should be in a sequence of times was dependent partly on that. And there was an estimation method presented, marginal structural models, and there were several assumptions. The treatment was measured fairly reliably. The measured covariates, even though they varied over time, were actually capturing the major sources of confounding, and the model of the treatment, given the past, is reasonably accurate given what the doctors really do. And the output that you got was an estimate of the causal effect with confidence intervals. So all good. The problem, or I think what I would describe as the fundamental problem of this sort of work, is that any estimation and any inference you're doing is conditional upon the model you've specified as being accurate, right? And that includes all the parts of the model. So as Phil Stark presented earlier in the day, right, using all different kinds of tests just as a simple check to see if a treatment versus control, right, t-test, paired t-test, or permutation test, that's another way in which the model might be specified correctly or incorrectly. And the inferential statistics depend crucially on that. So what you're up against when you're doing estimation and inference is all the other parts of the model, right, which govern how good the inference is. So if you're doing search, it's a completely different context. So take a question that was answered in a nice piece of work in 2012 about regulating flowering time in a plant called Arabidopsis. In this case, they had very little prior knowledge of what genes would actually regulate flowering time, and there were 25,000 candidates, right? So you can have a lot of money to do a lot of experiments, but you can't do 25,000 experiments and hope to actually have anything like knowledge in the immediate future. So in this case, we did a graphical model search, and what we had for measurements were RNA microarray data, and we had to make an assumption that RNA microarray measurements gave us a reasonable proxy for gene expression. We had to make all sorts of other assumptions that I won't go into, but we're happy to talk about and make explicit. And then the output was not pulling a rabbit from a hat, but it was a series of suggestions for candidate genes that might regulate flowering time, which we could then follow up with experiments where we knocked out those genes and figured out whether or not they actually did affect flowering time. Okay, so besides the assumptions, which are definitely important to make and incredibly controversial in some circles, the fundamental problem with model search is that the number of models to search over grows super exponentially with the number of nodes in the space. So here's a depiction of how many models, right, even eliminating cycles and not including latent confounders, there are if I have 1 through 10 variables, right? Now, 10 variables is a tiny space, and when you're talking 25,000 genes, you're talking 25,000 variables, so you can see that if you grow super exponentially, you're going to have so many models that it's not even possible to think about searching over all those models. You have to have some very intelligent, efficient search algorithm to get through that space and have any hope to come up with something reasonable. So in a way, I think this paradigm conceives of the basic enterprise of causal discovery is you have data, you have prior knowledge, all right, and that prior knowledge can range over what kind of variables you have, whether there's plausible causality, what sort of measurements you made, et cetera, et cetera. And you have a whole variety of analysis techniques, right, some human, some computer, some both, from which you actually get networks. And then from those networks, well, I should say, first of all, the data can be experimental and observational. From the networks, you have to generate what causal hypotheses you think are interesting. And that can be done in a myriad of different ways, by humans, by computer, and by both. And the main goal of the research that we've done is to give you a really well-studied, theoretically motivated computational aid to suggest plausible hypotheses, which you can then test by experiments. And then once you have the experiments, you can actually gain more data and more prior knowledge and iterate infinitum. Okay, so what are the basic components that you need to learn anything about causal networks from data? You need a representation of causality, right? You need actually a way to evaluate which causal models fare better against the data than others. I need a way to search those space of models like I told you before. So the model representation, right, is the simplest possible one you could imagine. It's causal graphs, which are entirely qualitative, that have no quantitative information whatsoever, and they simply represent which variables directly cause which others, right, relative to the system of variables you're talking about. So here's the simplest example I can think of, right? How much television one watches, right, might, in the end, have an effect on body mass index, and I'm not claiming this is the true causal model, by the way. But if it's going to get to obesity or BMI, the mechanisms or the pathways that might go through tend to be thought of as either it's going to make you exercise less or it's going to make you eat more, right? What's notable about this graph is that there is no edge from TV to BMI directly, and that has to have some consequence that you could test or actually motivate as having a meaning beyond just I didn't draw the arrow there, right? Now, contrary to what Phil Stark was saying, the whole purpose of putting a representation like this together and then connecting it to statistical constraints was not to redefine causality, but was to be indifferent about it. You pick your own idea of what causality is. It could be potential outcomes, it could be interventional in a different way, it could be probabilistic. What we need is some way to connect, right, the causal structures with something you can test in the data. And what we claim to have done is found a connection that anybody can agree on, no matter what your definition of causality is. So if you go a step further and you actually say, I'm going to parameterize these graphs with probabilities or numbers, then what you have to do is give for each variable that's in effect what its distribution of probability is given any possible value of its causes. If you've done that, you are in a causal Bayes net. And here's an example of one that was used in biomedical research. Karen Sachs did a study on single cell activity in genes and proteins and came up with this network using some of the methods that I'll talk about over 10 years ago. So there's lots of model representation issues, right? In some cases, you can assume you have no prior knowledge about anything about the networks. You can assume you have deterministic, you can have probabilistic relationships. The variables can be discrete, continuous, or both. You might have no time dynamics. It might be a cross-sectional study. There might be time series. There might be discrete time continuous time, etc. Distributional assumptions have to be made. Feedback can be treated as absent or present. Latent confounders can be treated as absent or present, etc., etc. So there's been a myriad of contexts in which different kinds of assumptions have been studied in terms of what we can actually learn and what we can't learn from those different contexts. Okay, so all this depends upon having some way to evaluate, right, the causal structure and see if it actually fits the data or not. And the relationship, right, that's driven a lot of this field, right, you can express as either the causal Markov assumption or this dependent separation relationship that Judea Pearl and his students discovered way back in the 1980s. There's a whole discussion to be had about whether or not causal graphs entail these kind of probabilistic constraints by deseparation of the Markov consumption. I will fully go to the mat and say that every one of you, if I had enough time, would agree that yes, this is a perfectly reasonable assumption. It's not controversial. But in this simple case, if I have a causal graph and it says that X is a cause of Y is a cause of Z, but X has no direct influence on Z, and there's a minimal completeness assumption, in which case you're saying, I don't leave out confounders, this is all the action there is, then that, by deseparation, entails a testable statistical prediction. And I'm totally non-parametric now. I haven't put any numbers in here. It just says, if this is the qualitative causal structure, then that's the conditional independence relation I should see no matter how I parameterize this model. So that's model evaluation. The models are going to entail constraints. We can look at the constraints, see if they hold. We can look at simultaneous set of constraints and see if they hold jointly. Right? There's lots of statistical ways. You can do a Bayesian score. You can do it all sorts of other ways. There's a million ways to evaluate models. Model search is going in the opposite direction. I start out with something in the data that I could test. Right? It doesn't have to be exactly conditionally independent. It has to be approximately so. And then I could say, well, what's the algorithm going to do? It's not going to search for, right, what particular causal DAG, right, explains those set of constraints. That's another misconception that's persisted for decades now. It's going to tell me what are all the causal models that would explain that set of constraints. Because if I don't assume anything more, I can't learn anything more. I don't know anything more. All I can tell you with this minimal set of constraints, and that is the thing I observe in the data and inferred holds in the population, and the assumptions that connect causal structure with probability, is that it's one of these three graphs. But they're all equivalent unless I do more work, more experiments, more parametric assumptions, et cetera, et cetera, et cetera. So all we can really do with any of these computational discovery algorithms is start with data and then compute the set of equivalents, right, an equivalence class of models that we could not distinguish among. And that tells us, in some clear and very, very rigorous way, the extent of what we could know from that data unless we did more testing and made more assumptions. But before I move on, I should say that what you are trying to do in many of these cases is see, well, if this is the whole set of models that the data seems to support, then if there's something in common with all those models, then I can actually say I've learned it, right, from the assumptions I've made and the data I've gotten. So in this case here, right, I have no idea from this data of the direction between any of the two variables x, y, and z, right? In each model, there's a difference in the way the edges are oriented, going one way or the other. But what I can learn, right, if the assumptions are satisfied, is that there's no direct relationship between x and z. That whatever the relationship is, it goes through y. So those are the kind of things, right, that one gets from, right, the output of the algorithms. Okay, so there's two sort of sources, there's two strategies for doing model search that have evolved. One is called constraint-based, and it relies on individual tests of constraints or implications, and then paring down the search space based on individual test results and doing it very efficiently. And the other is a score-based approach where I take the model and I say, well, what score can I give it? How well does it explain the joint set of constraints I observe or don't observe in the data? So let me give you examples of each, and I'm going to completely wave my hands and just literally sketch these things because it's much too much to go into. But I do want to illustrate it on a couple of cases and show you its utility. So in the constraint-based approach, I look at the constraints that hold, and then I use the pattern of constraints to narrow the space dramatically each time I do a thing. Okay, so here's an example I referred to briefly before, which is the flowering time of Arabidopsis thaliana. So the question was, which genes regulate flowering time, right? And that was measured as the days to bolting. And with the aim, of course, was to get plants that flower faster so we could sell them quicker. And it's not we, this is all done by a different group than my group. So the data were 47 plants, which varied on their flowering time, and each plant was put into a microarray, and so we had data on about 21,000 genes. So we have this data on the genes, and that expresses for each little dot how much of the gene is expressing itself in those 47 plants for each of the plants. And then we applied a constraint-based discovery algorithm, and from that we got about 25 candidate regulators of flowering time. And then we went back to the lab and we see which of those candidates actually produced different flowering times. So there were 25 genes in the final list. It turns out 5 of them were known regulators of flowering. And of the 20, it turns out luckily there were 13 that were actually, we had seeds that either knocked those genes out or actually up or down regulated them. And then of the 20, of the 13 that we actually got to test in the lab, 9 yielded replicates that survived. So we could actually see if the flowering time differed. And it turns out that 4 of the 9 actually affected flowering time. So and this is more complicated than it should be, but the basic chances of identifying a gene regulator before you did any of this work were about 1 in 1,000, right? And now we've reduced this to approximately 1 in 2. So have we found causality? Definitely no. Have we done a lot to actually get ourselves candidates that are causal? Yes. And we tested a lot of other techniques, regression-based techniques, correlational techniques, machine learning techniques. All these techniques that didn't rely crucially, right, on the deseparation relation that's a signature of causation, right, performed no better than chance. So it's not just taking advantage in a clever way of the associations. You have to actually have causal information to get this to work well. Okay, so the score-based approach is you take each, not model, but equivalence class, because you have an assumption, right, that every member of the equivalence class is going to look the same. So what you're really searching for is not a model, it's you're searching over an equivalence class. And then we do a greedy hill climb search, and we've speeded that up lately so that a popular learning algorithm called greedy equivalence search, GES, that was essentially limited to about 100 variables or less, right, has now been optimized so it can actually handle up to 1 million variables. So we tested it in simulation by generating random Bayes nets, random models, each of which had 30,000 nodes and about 60,000 edges, which actually makes it very easy compared to a much, much fuller graph to find. And we sampled every one of those to generate 1,000 cases, and then we looked at the results of applying the algorithm. Right, we could do it in about 2.5 minutes on a supercomputer and using 128 processors. And the average precision was 99% and the recall was 84%, which means the algorithm was a slightly conservative. So when it actually output, right, an edge or an equivalence class that had a connection, right, 99% of the time it was there, but it didn't get all the ones that it could have. Okay, so we applied this in a second case, which is really looking at how the brain is connected. So you are all pretty much familiar, I would think, of what fMRI studies do, right? They put you in a magnet, and then you get voxels like in your brain measured for how much oxygen is flowing through them, and then from that inferring how much activity is going on neurally in those little voxels. And we can now do about 44,000 voxels in your brain in any given study. And as you can see from the picture, some of the voxels look like they're very active and some are not. And so there's been lots of different things made out of what brain activity, what brain regions are active and what brains are not. In these kind of studies, the neuroscientists worked very hard to identify what they call regions of interest. So instead of analyzing 44,000 voxels, they would collapse those regions into maybe 10 or 20 different regions which were taken to be something functional in your brain. And then we'd run this data, and there's lots of pre-processing I won't go into, through a score-based causal discovery algorithm, And what we get is the causal connectivity among the regions in the brain when they were processing a particular task given to the subject in these magnets. And that's been published in a whole bunch of different places. But the thing I want to show you, and is I think interesting to note, is that this was actually applied to determine the difference between, or detect the difference between autistic and neurotypical subjects, right? So the usual approach for that is to search for differential recruitment of brain regions. Well, that turns out to be very, very difficult in neurotypical versus autistic subjects. They each have lots of active regions in the same basic way. So instead, this group at Rutgers took an approach where they gave subjects 3 tasks: face processing, theory of mind, and action understanding. And then they actually tried to infer the causal structure of the network among these regions of interest for each of the tasks. And then compared how your brain was processing, right, if you were a neurotypical, and how your brain was processing if you were an autistic spectrum subject. So the results were really interesting. I can finish in 5 minutes. So for the face recognition task, the networks were very similar, although you can't see the little numbers attached to these edges, they're not too different either. So there was no differentiation between subjects who were neurotypical and autistic spectrum on face recognition. On theory of mind, the results were striking, right? We have a substantially different network among autistic spectrum subjects and neurotypical subjects. And then if you do a theory of action, right, there was differences, but they were less. So the basic outcome of that is that even though you can't see differences in these subjects about how much brain activity is happening in different regions, you can see quite a difference in terms of the causal structure of that brain region communication. Okay, so let me just finish by saying that all this work that's happened over the last 20 or 30 years, which really originated in computer science and has lately sort of found its way more into biomedical research, is now going on in a Center for Causal Discovery that was funded by the National Institutes of Health 2 years ago, and that combines work in bioinformatics at the University of Pittsburgh, computational work in CMU, the Pittsburgh supercomputer, and a group at Yale doing work on biomedical discovery. So this is one of about 12 big data to knowledge centers the NIH funded across the country about 3 years ago. And the purpose of the center is to develop new discovery algorithms, right, that'll actually be useful to biomedical scientists to make these tools widely available and to attack with these tools 3 actual research areas. So the driving biomedical projects are discovering cell signaling networks in cancer, right? We've heard something about that today. And I was going to present a case study from cancer, but when I talked to my colleague Greg Cooper at Pitt, he says, well, it's not yet published, and so I'm not allowed to show you the results, or then they can't publish it. But that's exciting stuff coming up. The second research project is to discover the mechanisms of disease onset in lung, chronic obstructive pulmonary disease, and pulmonary fibrosis. And the third thing, which I gave you a very quick illustration of, is to discover the functional causal connectivity of regions of the human brain from fMRI data. So in summary, I think causal search is extremely important to actually study and improve in situations in which there's a large number of plausible hypotheses. We have weak prior knowledge, and the idea of actually estimating or testing or experimenting on particular pathways or particular hypotheses would be impossibly slow and long. And I think it's clear to say that we now have search algorithms that are provably reliable in the large sample limit, given the assumptions that are made vivid, and that are actually useful in suggesting causal hypotheses that were later confirmed in many fields now: biomedicine, economics, public health, educational research, and many other domains. And also, I think it's important to note that unlike what was out there in the early 1990s, these algorithms now cover a very wide variety of research contexts. systems with latent confounding, systems with feedback, time series, nonlinear systems, psychometric measurement models, just to mention a few. Thanks.